Now we're going to take a look at contrasting the process by which enzymes help reactions to proceed with ATP. If you're not sure that you really understand the process by which ATP catalyzes reactions, I strongly recommend taking a look at the Khan Academy tutorial, which was found in the previous video and posted on the course website. But here's an opportunity for you to test yourself. Pause the video here and tell me which of these describes why ATP is able to drive chemical reactions. Hopefully you recognize that this is the process of energy coupling, the transfer of free energy released when ATP is hydrolyzed to ADP to the reactants of an endergonic reaction. Enzymes are going to be responsible for speeding up metabolic reactions. This is different than what ATP does. ATP takes an endergonic reaction, a non-spontaneous reaction, and makes it spontaneous. It says nothing about the speed. Well, enzymes are going to take a reaction that's already spontaneous, and they're going to reduce activation energy to increase that rate. It's because of this that we call them catalysts. Enzymes are catalytic proteins, and they're going to be important for driving each one of the reactions in a metabolic pathway. Now, I do want to drive home one point. We're going to be talking about enzymes in a very general form, but enzymes are actually very specific. They can only catalyze a specific reaction. Even when you talk about an enzyme having a particular substrate, more on this in a minute, that can only deal with one of the possible reactions for that substrate. Enzymes are very, very specific, which means that your cell has many, 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 many different enzymes for all the many, many, many different chemical reactions that make up its complex metabolism. Enzymes are going to be able to speed up reactions by lowering the activation energy, which is really a barrier between reactants and products. Even for spontaneous reactions, there is going to be an amount of energy that's necessary to get through that transition state. Enzymes are not going to change the actual difference in free energy, meaning enzymes aren't going to do anything that actually releases energy into the system. You're going to have a set energy for your reactants and a set energy for your products. The only thing that they will do is reduce that amount of energy that's required for the reaction to proceed. So they're capable of speeding up a reaction that's already spontaneous. So take a look here and take a look at my reaction curve. Select all of these which apply to the difference between these two curves. Well, you've already looked at how ATP is going to influence a reaction curve. It adds energy to reactants, but in this curve, there's no change in the energy of these reactants. The black curve may occur at higher temperatures. It may not. We don't really know. There's nothing here that identifies that for us. But we can say that the red curve has a lower activation energy, and so it's going to be catalyzed by an enzyme. So which of these describes why enzymes are able to drive chemical reactions? Good. Enzymes reduce the activation energy of that reaction. So true or false? An enzyme is capable of making this endergonic reaction spontaneous. Well, this would be false. Enzymes are only capable of reducing activation energy. What makes the difference between a non-spontaneous and a spontaneous reaction is going to be the relationship between reactants and products, the amount of energy that is in those reactants. Well, the only thing you know that can change the amount of energy in reactants is ATP's energy coupling. So if a cell had an endergonic reaction like the one you see here that it needed to occur, it cannot use an enzyme to cause this reaction to occur. What would need to happen is energy coupling and the hydrolysis of ATP, which would make this non-spontaneous reaction spontaneous. If an enzyme were to come in and assist the process, it could cause this reaction to occur faster once ATP has gone through and added energy into the system. Remember that enzymes are very, very specific. This is different from ATP. ATP is hydrolyzed. It's the same ATP undergoing hydrolysis. But enzymes are really what help to pick exactly where these things occur. So let's say, for example, you have an endergonic reaction that you want to occur, like the formation of glutamic acid. Well, if we wanted to go through the process of forming this, you would use an enzyme to not only couple the hydrolysis of ATP to a specific reactant, but you could use that to help you lower the activation energy and get to your products. The reactant that an enzyme acts on is called the substrate, and that substrate is going to interact with the enzyme at a specific location on that enzyme known as an active site. It's the region of the protein where the substrate or substrates bind. In this process, which I'm about to show you, you're going to notice that the enzyme is not consumed. This is very different from ATP, which is consumed by the process of hydrolysis. It is broken down, cannot be used again. Enzymes are not going to be consumed. There's no energy that's stolen from them to help the reaction proceed. And so they're going to be able to have many, many, many catalytic events. A very small amount of enzyme is going to be able to do a lot in the cell. So substrates are going to bind the enzyme at active sites. That active site is going to lower the activation energy. It might destabilize bonds that are needed to break, stabilize bonds that are needed to form. Perhaps it's going to have a particular pH or bond types. It might be a hydrophobic or a hydrophilic region. These are going to be conditions that encourage the substrates to be converted into products. That's what we mean when we say activation is lowered. It creates favorable environments for the reaction to proceed. Those products get released, and when they do, that active site is now empty and available for new substrates to bind and the process to repeat itself. Remember that enzymes are proteins, and so like all macromolecules, their function depends on their shape. Temperature, pH, chemicals in the environment, these are all examples of things that can influence enzyme activity by affecting the protein structure. Shown here is an example of two different proteins at two different enzymes and the pH that's necessary. Pepsin is a stomach enzyme, while your stomach is a very acidic environment. And so pepsin functions best at the pH of your stomach. So at about a pH of 2, pepsin is going to have its greatest effectiveness. But trypsin, which is an intestinal enzyme, functions at a near neutral pH of 8. This is about the same as what you would find in your intestines. And this is because the differences in pH cause slight changes in the structure of these proteins. The protein that forms trypsin is going to be at the right shape to be able to do its catalytic job at this pH of 8. But at a pH of 3, trypsin is not going to have the same structure, which means it can't carry out its function. Pepsin is going to be a polypeptide, a protein that folds correctly at a pH of 2. But at a pH of 6, pepsin will not have the right shape. The regulation of enzymes, so the intentional regulation by a cell, is going to be critical for the ability of the cell to control all those many different chemical reactions that are occurring inside. It'd be chaos if every enzyme in the cell was all acting at whatever rate and whatever processivity that it wanted to all the time. No. This is all tightly regulated and controlled. We're going to talk about 3 different ways in which the cell can regulate enzymes and thus regulate metabolic activity. The first is a competitive inhibitor, which does exactly like it sounds. A competitive inhibitor is simply a molecule that binds directly to the active site and outcompetes the substrate. Now, what if I were to draw you back? Remember that enzymes are proteins. Proteins function based on primary, secondary, tertiary, and quaternary levels of structure. So the interaction between multiple polypeptides would be known as which of the following? Tell me on Socrative. Good. That would be quaternary structure. And just to drive the point home, pentary structure is not actually a level of protein structure. Most allosterically regulated enzymes will be made using quaternary structure. Allosteric regulation is a regulation that controls shape. So competitive inhibitors are responsible for outcompeting a substrate. Allosteric regulators are going to either activate or inhibit a protein by influencing its shape. Most commonly, the quaternary level of protein structure and shape. So if you have an allosteric inhibitor, it's going to bind to your protein and cause that protein to be stuck in such a way that it's not active. If you have an allosteric activator, you can activate the protein by stabilizing the active form. This is a great example of how we can turn things on or off. And as a matter of fact, you can have proteins that are constantly competing and switching from one form to another. The last level of regulation is known as feedback inhibition. Feedback inhibition is going to be very similar to competitive inhibitors, but with one very important difference. And that is that the product responsible for outcompeting the substrate is actually the end product of the pathway. It's a feedback loop, right? When you get to the end of the semester and you fill out a survey about teaching, you're giving your teacher feedback. It's the end product of the semester, and you're giving feedback at an earlier step. Well, metabolism can work the same way. The end product of that metabolic pathway can provide feedback earlier in the system. In this case, by inhibiting an enzyme that controls earlier in the system. These are most important for when you want to limit the amount of end product produced. If you already have an abundance of the end product, the easiest way to shut down the system is to turn off one of the enzymes which is necessary for its synthesis. Now what I'd like you to do is to watch this video posted on the website about how caffeine keeps us awake. Not only is this going to be a nice little introduction to ATP and showing you some protein functions, but this is a little bit of a preview as well about some of the things that are going to be coming up in this class, especially when we look at cellular communication. Once you've done so, go ahead and answer my Socrative question about how it is that caffeine keeps us awake. And as you're reviewing, I would like you to test yourself with one final question, a short answer. Tell me in Socrative, why have cells evolved to use both ATP and enzymes?